Our interest of the Max Planck Institute in the mining impact study is to study biogeochemical processes, ecosystem functioning and microbial diversity in the mining impact areas. We use two different approaches to study these aspects. One is to do in situ investigations of microbiological processes like oxygen fluxes in the sediment, and the second one is on the ship in the lab to do biodiversity sampling of the microbiological community. To do the in situ measurements, we use three different kinds of ROV modules which are operated by the ROV. So we bring down the instrument with an elevator system to the seafloor, deploy them with the ROV at the distinct areas where we would like to do the measurements, and then we perform the measurements. One is to do oxygen microprofiling in the sediment using two types of profiler. One is with electrochemical sensors to do shallow profiles of 15 centimeters, and then we have a new optical fiber profiler which can penetrate into the sediment up to 30 centimeters. With these profiles we can measure the activity of the microbial community at the seafloor. The oxygen microprofiler works this way, that we have a small sensor of a tip diameter of 50 to 250 micrometer which penetrate in small steps of 0.0 millimeter into the sediment to record the oxygen concentration from the overlying water through the sediment water interface in the sediment. From these profiles we can then calculate later on deck the oxygen flux, means the respiration activity of the microbial community. Oxygen profile gives us a good measure of the activity of the microbial community and gives us an overall activity of the seafloor community. So the first indication and a fast and reliable measurement of the activity. And this is also important for the mining impact. So if we get a disturbance in the sediment, we can easily detect the difference in the activity of the microbial community which sits directly at the sediment surface. The other aspect of the microbial community activities we study here on board is the community distribution and the abundance of the microorganisms. So we take sediment from the different locations and analyze the abundance of the microbial community and the diversity to see how this changes in the impact of a potential mining activity.
So here on board we are interested on bottom water, on sediment from the deep, so from 50 centimeters to 5 meters. We got push core samples from the RV. We also got muck samples, so muck cores, so they were around 50 centimeters long. We got gravity core samples, so these are samples, so sediment cores, they are 5 meters long. And we also got box core samples for manganese nodules. And we also had a lot of CDD casts for water samples, so bottom samples, so 5 meters above the seafloor, 25 meters and 50 meters above the seafloor. When the sediment samples are on board, we go into the cold room and then we slice the sediment cores into different layers. And then we take RNA and DNA samples for community studies. So we will sequence the DNA and RNA back home in our labs. And we also take samples for other parameters, for example, for organic carbon and porosity or chlorophyll. But we can also not detect all of these parameters here on board, so we have to do that at home. We also took sediment samples from manganese nodules, so we crushed material from the surface, from the bottom, and we also cut the manganese nodules into some pieces, and we tried to scratch some materials out of the manganese nodules. So when there is deep sea mining, the upper sediment layers of the deep sea floor is removed or changed, and also the sediment plume can change the sediment structure. So therefore the sediment community of the upper few centimeters and maybe also meters can change, and this can have a huge impact on the microbial community itself and also on the biogeochemical cycles.